signal transduction. فالمشكله الطلاب بضلوا ساكتين، يعني ما بيحكوا مثلا هاي اخذناها هون وهي اخذناها هون. هلا بالنسبه لهم كله بضل ماشي وبعدين بفلتروا. فبدي حدا يعمل فيدباك عن اللي اخذناه، انه هذول ان اخذوا هون وهون كتوبكس، تمام، مشان نعمل يعني مقاصه انجاز التعبير، بحيث انه نخفف محاضرات البيوكيمستري. هلا للامانه كانت هي 4 محاضرات للبيوكيمستري، بس اجى حدا من خارج الجامعه، اتوقع دكتور عبد الفتاح الحادر، كان بعطي الفيزيولوجي، فنقل محاضرتين للبيوكيمستري، صارت هاي الكونفليكت. ولا احنا ما بدنا اكثر من 4 محاضرات كبيوكيمستري للامانه. طيب هلا راح نحكي اليوم عن ايش القصه. الليزر. طيب راح نحكي عن الهرمون اكشن والسيجنال ترانسدكشن، اللي هو برنسبل مكرر كثير، بس راح نحكي عن سبيسيفيك توبكس بهذا الموضوع. شابتر 42 من هاربرز تكست بوك is the reference for this lecture. So basically كلنا بنعرف انه الهرمونز بغض النظر عن الشكل تبعهم والتارجتس تبعهم eventually they will transduce signals، وهاي السيجنال transduction process ممكن تصير من خلال ملامسه الريسبتورز او الارتباط بالريسبتورز اللي على سطح الخليه، او بالسيتوسول، او بالنيوكليير 1. حد الان ما سمعناش عن ريسبتورز لهم علاقه بالريجوليشنز تبعون الميتوكوندريال جينوم فور اكزامبل. Okay, and how they are going to be regulated by signal transduction, which is something very interesting to dig. So the stimulus itself is kind of challenge and sometimes could be threatening to the organism itself إذا كان هو unicellular organism على سبيل المثال. Okay, as a stimulator, the organism itself can deal with that as a threat. إذا بنحكي عن unicellular organisms زي البكتيريا، تمام. So sometimes they respond to that by avoiding exposure to that thing. فعشان هيك هذا عبارة عن نوع من الثريت rather than signaling process. هذا بينطبق على الكائنات وحيدة الخلية. Okay. لأنه مثلا ممكن يكون radiation، ممكن يكون أي نوع من التوكسين in the environment. It could be biological factors. كل هذول ممكن ياثروا على هاي الخليه. فلما نحكي عن الثريت هون بالنسبه للاورجانيزم نحكي عن اللو ليفل اورجانيزمز rather than الهاي ليفل اورجانيزمز. الاورجانز themselves، لانه كل اورجان يعتبر اورجانيزم مجازيا من ناحيه استقباله للسيجنالز. So this is a challenge or threatening sometimes, which means if it's threatening, so the tissue or the organ is trying to respond to that by either reducing the toxicity or developing resistance. خليني أعطي مثال معاكس تماما للنورمال situation. For example, بالcancers we use different kind of treatments. من ضمنهم الكيموثيرابيز. Chemotherapy, you are using chemical compounds to treat the cancer. هلا some types of cancers they develop kind of resistance to that treatment. So it looks like as a smart organ if you would. بس هو فعليا مش سمارت بالمعنى الحضور الذهني او ذهنيا. انه في عندنا ريسبونس داخل الكانسر تيشو بخليه يقاوم هذا النوع من التريتمنت اللي هو بالنسبه له ثريت كتيشو او كاورجان. The integrity of signal or a signal cell within that organism is also another challenge, which means that you have to keep kind of homeostasis when you respond. This is the meaning of integration or integrity of the cell signaling or signal inside the cell. And this homeostasis means the signal pathway لازم تكون regulated بمعنى انه it will not be out of control. لأنه otherwise it could be pathogenic or cause pathogenesis, or at least it can be just something related to metabolism or defect in the metabolism. فهذول الاشياء بحاجه انه نعمل شيء او يكون شيء داخل الخلايا هذول او على هذول الخلايا يعمل transduction للسيجنال to avoid response بطريقه انه يكون sometimes lethal or pathogenic. هلا احنا بنعرف كل انواع الستيمولس سواء كانوا هرمونز or other than hormones، لانه هنا بتعامل مع اشياء ممكن تكون lights for example they can do stimulation for certain organs rather than the hormones. مش بضروري يكون molecules. لما بنحكي molecules معناها hormonal action خصوصا بالاندوكراين سيستم. بس الستيمولس could be other things, could be for example light or certain kind of wavelength or X-ray إذا بدك. كل هذول عبارة عن stimulus and they are sometimes activating two kinds of hormones. مجموعة بسموها المجموعة الأولى group 1 and group 2. هلا الجروب 1 اللي هم مصنفين ضمن الهرمونز اللي لهم receptor. So if they are forming complex بالbiochemistry they are classified to be group 1. Group 2 many different signals that is not involving complex formation with the receptor. وبالتالي ممكن الهرمون نفسه can do the action itself by activating certain pathway and we have all of the pathways inside the cell like protein modification, protein translocation, transporting channels, gene transcription. فكل هذول ممكن الهرمون نفسه يروح يؤدي الغرض او يعمل stimulation لهذول التارجتس. على سبيل المثال ممكن يكون الهرمون نفسه transcription factor that will bind the DNA and causing either overexpression or down regulation. هلا other hormones require receptor عشان يروح يرتبط بالدي ان اي if the target is the DNA. That's why they are classified to be class 1 and class 2. هلا signal how it's going to be generated by the binding of the ligand. Simply if the ligand binds the receptor or binds the target, الليجند اللي هو الهرمون بقصتنا. So if it binds the target it will cause stimulation or اللي هو stimulus itself and generation of the signal. So first of all you have to have ligand receptor complex in order to have signal in group 1. So generally group 1 زي ما حكينا قبل شوي مشان نتذكر عن ايش بنحكي انه هذول requiring certain receptors in order to transduce the signal. So as an example thyroid hormones and retinoic acid for example they go directly to the nucleus because they are hydrophobic molecules and of course they need certain kind of transporters if they are not if they are hydrophobic molecules. So they pass through the cell membrane, nuclear membrane until reaching certain kind of receptors. And in this case, the receptor itself is transcription factor or a factor that will bind the DNA that will bind TRE region. هلا TRE هاي عبارة عن element or sequence in the DNA. اختصار لكلمة thyroid إذا بدك أو hormone response element. بحالتنا هاي لانه ثايرويد احنا بنسميه تي ار اي ثايرويد ريسبونس اليمنت اللي هي السيكونس اللي بتستجيب للاكشن تبع الثايرويد as an example. So before the binding of the ligand to the receptor itself اللي هو بحالتنا هاي transcription factor there will be inactive gene or inhibited. So the binding of the ligand or the hormone or the activator molecule will cause release of the inhibitor and activation of the gene until you reach the active situation and this will cause expression of the gene. هلا الاكسبريشن للجين هذا بغض النظر هو الجين شو راح يعمل بعدين، راح يعمل اكتيفيشن لشيء، راح يعمل انهبيشن لشيء اخر. We have response but this kind of response depends or relies on the path or the movement of the hormone or the triggering molecule to the receptor inside the nucleus. In some other occasions إذا كان التارجت هو DNA still some receptors are localized in the cytosol بالاصل موجودين في السيتوسول and they are in the inactive form بهذا الشكل. Okay, so the entrance of the receptor itself will cause dissociation of certain proteins like heat shock proteins in this case and binding of the ligand to the receptor that is localized in the cytosol and then it will be moved to the nucleus. So the binding of the ligand to the receptor will cause localization. Otherwise it will be relocalized in the cytosol. هلا هذا الشكل اللي هو الانactive form للهرمون تبعنا او للريسبتور سوري مش للهرمون للريسبتور and this one is the active one after the binding of the ligand to that receptor. So we have two options يمكن حكيناها بمحاضرة سابقة انه we have receptors inside the cell as transcription factors, we have receptors in the cytosol and they will be localized to the nucleus. Any defect in these receptors can cause kind of disease or disorder associated with the insufficient signaling or vice versa. It could be over signaling. Over signaling معناها انه في عندنا mutation بهذا الريسبتور that is always localized in the nucleus and causing continuous action. وهذا الشيء abnormal. So that's the meaning of integrity of the signal. It has to be regulated. هلا على مستوى الجين اكسبريشن او على مستوى الجين ريجوليشن هون بالنسبه للريجوليشن اوف ذا جين برودكت. جين اكسبريشن معناها في عندي جين برودكت. شو ممكن الهرمون يعمل حتى انه هذا الجين يصير اكتف اور ان اكتف حسب نوع الهرمون. It can control the transcription process or it can control the primary product itself اللي هو primary transcript ال premature messenger RNA. I mean, if you have the hormone and causing action, this action could be affecting the gene اللي هو المثال اللي هون expression or the stability of the primary transcript اللي هو المسنجر RNA before being messenger RNA اللي هو pre-messenger RNA or the process of modification or post-transcriptional modification بسموهم أو maturation of the RNA. So in either step you could control the gene by hormonal action. After that you also can control translation process as well as the stability of the messenger RNA outside of the nucleus. The protein itself could be modified. So the action of the hormone can affect any of these steps depending on what we know about that factor. اللي بنعرفه احنا مثلا كاكزامبل هذا انه في عندنا transcription factor that control gene expression for example. هلا الجروب 2 اللي هم البيبتايدز والكاتيكولامينز الهرمون have membrane receptors اختلفوا عن الاولانيين انه they cannot pass the membrane. Okay, so they have to have membrane receptors and these hormones most of them actually activates internal messenger if you would. وبحالتنا هاي في عندنا messenger or second messenger called cyclic AMP كاكزامبل لانه في عندنا many other types of second messengers. So the cyclic AMP is another intracellular signaling molecule and it's described as a second messenger that requires the action of adenyl cyclase in order to be generated. هلا activation of adenyl cyclase depends on the hormonal binding basically. So in order to activate this enzyme, in order to have this second messenger, we need the hormone action. Later on we have a lot of cascade reactions like activation of protein kinases or phosphorylation of proteins بسموهم phosphoproteins or phosphodiesterase as an example, phosphoprotein phosphatases اللي هم بشيل الفوسفات عن البروتينز. So all of these mechanisms could be activated after the adenyl cyclase action or activation in order to produce cyclic AMP. These hormones actually causing the activation of adenyl cyclase as an example اللي هو نوع من stimulatory hormones. So these hormones are classified as stimulatory hormones that rely on the activation of adenyl cyclase and cyclic AMP is the second messenger. هذول كexample. هلا others can be also cyclic AMP dependent but they are inhibitory hormones. They are inhibiting certain actions. That's why these hormones are classified to be inhibitory or بحطوا عندهم حرف I. هلا in order to activate الادينيل cyclase حكينا عن هذا الموضوع اعتقد بمحاضرة سابقة بس this is another demonstration. So in order to activate the adenyl cyclase which is localized in the membrane, you need the action of the hormone that will bind the receptor and you have to activate other kind of proteins. So this relies on the activation of what's called خلينا عليهم مباشرة شو it's here وين راحت؟ اوكي اللي هو G protein coupled receptor GPCR protein اللي هو 7 helical domains inserted in the membrane and this receptor is called G protein coupled receptor لأنه هذا receptor is coupled with a protein here اللي هو سميناه احنا المرة الماضية G protein لأنه بيعتمد على GTP. So eventually it will activate cyclic AMP and the activation of other signaling pathway inside the cell. So what I want you to understand is the following. إنه في عندنا adenyl cyclase that requires the action of the hormone that will bind the receptor and the receptor then will cause dissociation of the three subunits الالفا بيتا gamma. The alpha subunit is going to activate the adenyl cyclase and this is the pathway of the generation of cyclic AMP. كان حدا سألني عن إنه adenyl cyclase بيعمل conversion من ATP للcyclic AMP. طيب والguanylyl cyclase GTP. Yep. Okay. هلا بكل مره بنحتاج فيها ال ATP usually we have magnesium as a cofactor يعني بالreactions هذول. So the next thing is انه cyclic AMP هذا either continuously active راح يظل موجود or deactivated by the action of the phosphodiesterase. الكلام اللي كان مكتوب هون. وين رحنا احنا؟ سوري انا كثير احنا قلنا الادينين سايكليز activation will activate many kinds of proteins or enzymes inside. One of them is the kinase اللي هو الميجر one, and the phosphoproteins phosphorylation due to the action of the kinases. وفي عندنا هون الفوسفودايستريز. الفوسفودايستريز الدور تبعه feedback inhibition process. So ليش هاي الرسمه بتروح من عندنا؟ Yeah, this is the G protein. So the G protein is binding the hormone or the ligand, and then will cause dissociation of the G protein itself. This is the receptor, and this is the protein. And after the dissociation, the alpha subunit is going to activate the adenyl cyclase enzyme in our story. So here we have different types of G proteins, and they are coded according to their action, either stimulatory or inhibitory, or they are associated for certain factors like بسموهم alpha G proteins لأنه they are associated with alpha adrenergic effects اللي هم المركبات اللي بيعملوا اكشن زي الادرينرجيك افكت او الكولينرجيك افكتس. الكولينرجيك معناها بيعملوا اكشن مشابه للاكشن تبع الاسيتيلكولين or acetylcholine whatever. So that's why they are called alpha or alpha G proteins or G alpha proteins due to the action of these stimulus. So all of these are stimulators to these kinds of G proteins, and every kind is coded according to the type of the action downstream. هلا النوع الاول stimulatory usually activating adenyl cyclase and activation many other kind of reactions that will cause eventually activation of gluconeogenesis, lipolysis, glycogenolysis, or something related to the action of the cardiac muscles like the channels of calcium chloride and sodium. هلا الانهبitory one usually associated with slowing down certain processes like the heart rate because they are inhibitory, or neural electrical activity, or controlling the vision, for example, due to the response to the light. This is an example of the non-hormonal stimulator اللي حكينا عنهم. ممكن يكون اللايت للinhibitory G protein. So the point is that we have a lot of kinds of stimulators that could activate stimulatory or inhibitory or another stimulatory one, but it's a special type of stimulation process. This one is also stimulatory, but it has special mechanism. That's why it's classified to another or categorized in another class. So the point is, after the activation or the formation of the cyclic AMP, it's going to do its action by activation of certain kinases. في حالتنا هي عندنا بروتين اسمه protein kinase A. It's in active form. While the cyclic AMP is available, the regulatory subunit will be released from the enzyme itself. هذا الانزيم مكون من 4 subunits. اثنين منهم regulatory R واثنين منهم catalytic. So after the binding of cyclic AMP, the regulatory subunits will be detached and the catalytic subunits will stay together. So this catalytic unit is responsible for the phosphorylation process later on. That's why it's called kinase, and it's kinase A in this case. So many proteins are going to be phosphorylated. I'm not going to say they are going to be activated or deactivated, because the phosphorylation of the protein could activate or deactivate certain proteins. That's why we are saying its physiological effect or signal transduction process inside the cell. In order to have integrity of the signal, we have control, or we have to control the feedback inhibition or the inhibition of the forward reaction, which means that you don't always need phosphoprotein. So that's why the phosphatase should be activated by another action somehow. Here it will cause activation of the phosphatase. Phosphodiesterase enzyme is also going to be activated in order to deplete cyclic AMP. So these steps are regulatory steps for the starting or for the forward signaling pathway. And for sure, they are going to be regulated by the same action. I mean, cyclic AMP is going to activate a lot of proteins. One of them could do feedback regulation by activation of the phosphatases or phosphodiesterase in order to control the process itself. The continuity of the signal depends on the presence of the hormone. بمعنى آخر إذا راح الهرمون هذول molecules should be activated in either way. A cyclic GMP is also another second messenger as an intracellular signaling molecule, And we have many mechanisms that can form or associated with the action of cyclic GMP, like for example الاتريوبيبتينز اللي هو عباره عن peptides produced by cardiac atrial tissues that cause diuresis, for example, vasodilation, inhibition of aldosterone secretion. هذا كله له علاقه بالفيزيولوجيكال اكشن. نيتروبروسايد for example اللي هو عباره عن stimulators برضه للسايكليك GMP second messenger. Nitroglycerine. All of these molecules or compounds are associated with the action or the formation of cyclic GMP. Okay. The inhibitors for this is the phosphodiesterase. I mean, if you want to inhibit this second messenger, we have phosphodiesterases. One of them is the Viagra product, اللي هو commercial name for the sildenafil, or sildenafil, sorry, a drug. The action تبعه through the inhibition of the second messenger cyclic GMP اللي هو بيعمل vasodilation. Okay. Another special kind of signaling depending on the calcium, showing the importance of the calcium as signaling molecule. Indeed, the calcium itself is not that signaling molecule. It needs another protein or other factors in order to be signaling molecules. For example, there are three ways that the cytosolic calcium is changed inside the cell in order to activate the target protein, and then that protein will be the stimulating molecule. كيف تتحكم بالكالسيوم بالخلية؟ Either influx from outside, so it could be just stimulation of certain transporters that will cause influx of the calcium inside the cell, or some hormones indirectly promote calcium influx by modulating membrane potential. بمعنى آخر بعض الهرمونز بتخلي الكالسيوم يدخل لأنه هي الهرمون هذا بروح بيرتبط بالكالسيوم channel بيعملها influx. بعض الهرمونز indirectly they change the potential of the membrane, not the receptor itself. That will cause indirectly influx by stimulating the receptor or the channel. فواحد منهم direct وواحد indirect اللي هو بيعتمد على الاكشن of the hormone. And one more important source of the calcium اللي هو endoplasmic reticulum. So the calcium can be used as a signaling molecule, and that's why we need to introduce the calcium to the cytosol either from the external side or internal side. How it works? We have protein that's known as calmodulin. So briefly, calmodulin is single subunit protein, and it has this structure. There are 4 sites for the binding of the calcium, and upon the binding of the calcium to the calmodulin, there will be conformational change in the protein itself that cause binding of this calmodulin to the target one. So the action of the calcium as a signaling molecule is indirect one. It needs calmodulin in order to perform the action. فالكالمودولين هو اللي بيعمل السيجنال بس بيكون inactive لما بيجي الكالسيوم cellular activation and it will bind the target protein. طبعا this requires action of the hormone at the beginning. بالنهاية ما ننسى ان الهرمون هو اللي سبب either the influx of the calcium or release of the calcium from the endoplasmic reticulum. So calcium itself is mediator or is a mediator for hormone action. Mediator معناها اللي هو يعني transferring the signal between the hormone and the calmodulin. And they found that because the blunted action of the hormone by calcium-free media or when the intracellular calcium is depleted. They discovered if you remove the calcium or if you deplete the calcium from the media, the action will be stopped of that hormone. So they discovered or they concluded these hormones require or needs calcium as a mediator to have the action. Another approach ساعدهم على انه يعرفوا انه ليش الكالسيوم هو mediator can mimic by agent that increase cytosolic calcium. بمعنى الحالة الأولى انه كان يحط الهرمون بس كان يعمل depletion للكالسيوم، كان ما يحطش كالسيوم على الميديا. الحالة الثانية انه كان يحط اشياء بتشبه الهرمون وشوف الافكت تبعها على الكالسيوم. So in either way they discover the calcium is significantly important for the action or influences cellular calcium flux without the hormone indirectly by changing the membrane potential, for example. So by these approaches they discover that calcium is mediator and it's good or mandatory mediator for the action of certain hormones. These actions could include glycogen synthesis, pyruvate kinase, or carbohydrate metabolic pathways or energy metabolic pathways can be controlled by the calcium or calmodulin calcium mediating system. نيجي على الاكشن لاذر هرمونز like الانسولين and epidermal growth factors that binds the growth factors. So these receptors actually have similar thing اللي هو ability of tyrosine kinase or activity of tyrosine kinase. Tyrosine kinase means انه they have tyrosine site and they can do kinases for other proteins or can do phosphorylation for other proteins. So as an insulin receptor and insulin action, which is similar, very similar to the epidermal growth factor, this is what's going to happen. If the insulin is available, it will bind the receptor and this will cause release of what's called or phosphorylation of what's called insulin response substrate اللي هو عبارة عن substrate that's going to be activated by phosphorylation. So this protein basically is inactive when it's dephosphorylated and it's going to be activated after the phosphorylation, and this requires the action of the receptor itself. So the receptor has kinase activity that requires the binding of the calcium, and then it will cause phosphorylation of the other tyrosine in the target or the substrate of that modulator protein اللي هو inside protein. هلا later on we have a lot of signaling pathway ما راح اخذ في التفاصيل إلها because we have يعني bunch of signaling process. So just to know that we have this kind of tyrosine kinase mechanism in insulin or epidermal growth factor receptor, and this is the description of the whole pathway if you are interested to understand what's going on later on. So if you are just taking one example of these اللي هو cell growth, this protein for example, or a lot of proteins actually including these regulatory proteins are responsible for the cell growth and proliferation after the activation of the IRS اللي هو insulin response or insulin receptor substrate. So it's very complicated one to discuss, كل واحدة منها بدها محاضرة لوحدها. Another example اللي هو بسموهم JAK-STAT pathway because they have this kind of protein which is called JAK protein, and this is the receptor that is dimerized once we have the ligand, and the ligand could be any kind of these hormones like the growth hormone. And again, the autophosphorylation of the receptor itself is conducted once the ligand binds the receptor. بمعنى آخر إنه هذا بيشبه النوع الأول ببروسيس اسمها autophosphorylation mechanism. Autophosphorylation means that the receptor itself can do phosphorylation for the receptor itself. So later on, the activation of the receptor by phosphorylation will cause activation of many other downstream substrates like the STAT1, and we will have signal transduction inside. So this is just explanation of what the tyrosine kinases do actually by the phosphorylation of certain either autophosphorylation or phosphorylation of other proteins. هلا في mechanism خاصة بالtranscription process, and the question is how hormones affect transcription. أنا لخصتهم بهذول التيرمينولوجي أو بهذول الكونسبتس. إنه كيف الهرمون بيعمل الأكشن على transcription factor or how can the hormone activate or deactivate. إحنا حكينا عنهم بشكل مقتضب بس as stepwise there will be like the following. Actively transcribed genes are in regions of open chromatin area, which means if we want to regulate the gene area or the sequence has to be in a euchromatin form. بالجينيتكس في عندنا two forms للكروماتين structure, euchromatin, heterochromatin. هلا اليوكروماتين هي اللي بتحتوي على الجينز اللي can be accessed in order to be expressed. So first of all, they have to be at open area. Secondly, genes have regulatory regions in order okay. So first of all, it has to be opened. Secondly, it has to have regulatory sequences or regulatory regions or regulatory elements. كل هذول المصطلحات جائزين بالنسبة ل regulated genes. Okay. So if it's open and there is a regulatory region, the transcription factor can approach the target. hormone receptor complex can be one of these transcription factors, which means that زي ما بلشنا المحاضرة إنه you could have a hormone with a receptor as a complex that could approach target, or it can be in either way, سواء cytosolic or nuclear one. Or alternatively, other hormone generated signals can modify the location and amount activity of transcription factors. الفرق بين النقطتين هذول إنه الهرمون can approach transcription factor and then approaching the gene in order to do regulation. The second one is the hormone itself causing signaling that will eventually generate certain factors that will approach the transcription factors. فالفرق بينهم انه the first one is direct and the other one is indirect approach. Member of a large superfamily of nuclear receptors act with or in a manner analogous to the hormone receptors. So we have what's called hormonal analog or hormone analogs that can act instead of the hormone itself or even the mediators of the hormones. So we have a lot of analogs inside the cell that belong to a large family of nuclear receptors. These nuclear receptors interact with another large group of co-regulatory molecules. So both of these are talking about the binding of analogs to certain intracellular receptors and then approaching the gene rather than the hormone itself. So they belong to the super family of these hormonal system. ال HRB أو sorry ال HREs هذول كلهم عبارة عن hormone response elements, عبارة عن sequences that is found in the genes in order to be approached by the regulatory proteins اللي هم ممكن يكون transcription factors, and this is how it's going to be regulated. For example, we have these regulatory elements, androgen or hormone response element, whatever the kind of the regulatory elements. So it needs this kind of conjugation between the receptors and the ligand here in order to make stabilized form for the RNA polymerase that's going to cause transcription of that gene. So the transcription process is stabilized or stimulated or sometimes inhibited by the action of the regulatory protein or the receptor of that protein, but it requires the presence of these regulatory response elements or hormone response elements. So the action of the benefit of having these regulatory elements is because we have this ability or the ability of these hormones in order to make this stable conformation. بلغة عربية بسيطة جدا، حتى انه هاي regulatory elements تمارس الدور تبعها في وسيط بين regulatory element وبين promoter of the gene that is manipulated or regulated by the action of the receptors and the ligand. هلا الريسبتور هذا اذا كان هو nuclear one or cytosolic one transferred inside the nucleus, it's the same effect at the end of the day. هذول عباره عن نوع من الفاميليز للريسبتورز اذا بدك، مقسمين حسب نوع الريسبتورز. النوع الاول بسموه steroid classes or RXR partnered receptors or orphan receptors. الفرق بينهم انه الستيرويد requires steroid hormones. This requires other kinds of receptors other than the steroid one. Orphans means that you don't have ligands for that receptor. So in all of them you have to have dimerization. هلا هون في عندي ligand للريسبتور، في عندي ligand للريسبتور. Here we have no ligand. So these receptors are going to be activated just by dimerization and causing signal transduction by just making what's called homodimerization. هلا ال RXR يعني مشهور بانه بيعمل شيء اسمه heterodimerization. It means that you have to have two kinds of ligands, two kinds of receptors to do heterodimerization. In this manner, or steroid hormones, it's kind of homodimerization process. هلا الكوريجوليترز نرجع لهون. الكوريجوليترز عباره عن other proteins that are regulating the receptors themselves. Okay, one of them is called, for example, CBP اللي اختصار لكلمه CRIB binding protein. CRIB اختصار لكلمه cyclic AMP response element binding protein or protein. So we have this protein and we have another protein that can bind it اللي هو اسمه CBP. فهذا هو اللي بيرتبط في هذا. Okay, وهذا بيعتمد على cyclic AMP. ليش سموه كريب؟ لانه بيعتمد على السايكليك اي ام بي. طب شو بيعمل بعدين؟ له response element inside the DNA. So that's how it works. For example, if we have cyclic AMP that's going to activate certain kinases, so there are kind of response proteins inside the either cytosol or the nucleus that eventually bind the CBP, and it will cause activation of the signal transduction. So all of these eventually will reach the same target اللي هو CRIB binding protein that will bind the DNA. So this is the transcription factor actually that could be activated by many other kinds of signaling pathways. Depending on the signal pathway, we could have different actions inside the cell depending on the gene that will be available at that time. And that's the whole story. بشعر احيانا ان الامور تصل لمرحله الملل لانه we are just repeating what we said before. هلا اعتقد المره الجايه راح نكون خلصنا، لانه راح نحكي عن شيء بسيط جدا كلهم 10 سلايدز، and then نحكي عن الابروتشز اللي نستخدمهم بالمختبر ل diagnosis. That's it. يعطيكم الف عافيه.